Good afternoon, students. Today we are going to start with a very interesting topic. Now, first of all, answer a few of my questions. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Have you? Or why a mango seed forms only a mango plant and not any other plant? So you see, it is commonly seen that members of a species are largely alike. A horse resembles other horses and a rose plant looks like other rose plants. This is natural because only horses can produce more horses and only rose plants can give rise to more rose plants. Children resemble their parents, even grandparents or great grandparents. Sometimes, on seeing a newborn child, people often say he has father's nose, or mother's eyes, or grandfather's feature. So, this means that this resemblance among the individuals of a species has given rise to the apophism like begets like. The aphorism, in a way, implies continuity of life. It is not absolutely true that members of a species are seldom exactly alike. Children, also the two egg twins, often have some individual characters in which they differ from one another and also from their parents. In fact, their differences are as marked as their resemblances. Every living organism has a set of characters by which it can be assigned to a particular species. This is what we have seen in classification also in class 11th, and can be identified as a member of its own species, different from other species. That is how we classify the organisms, isn't it? So the similarities and differences among the members of a species, they are not coincidental, mind you. Just reflect upon this. They are not coincidental. They are received by the young ones from their parents. Now, which characters are received by the offspring from their parents and which are not received is a matter of chance. So, we come to heredity. Now, what is heredity? It is the transfer of information, that is, the characters, the resemblance, variations from one individual to another individual, that is, individuals of one generation to those of a future generation, commonly through the direct genetic descendants. That is what is heredity. So, I am going to read out the definition which is written here on your screens. Heredity, also called inheritance or biological inheritance, is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents. Through heredity, variations between individuals can accumulate and cause species to evolve by natural selection. This study of heredity in biology is genetics, right? So, the next point which we will take up is the genetics. So, heredity is leading us to the genetics. Now, genetics is a branch of biology concerned with the study of genes, genetic variation, and heredity in organisms. This term was coined by William Batson in 1906. Clear up till here? So you see the resemblance which is leading to heredity, genetics, they are interrelated. And now we come down to the basis of heredity. Firstly, what is heredity, and now what are the bases of heredity? We can divide the basis of heredity as physical basis of heredity and chemical basis of heredity. First, we'll take up physical basis. Gregor Johann Mendel announced in 1866 that heredity was controlled by particles called germinal units or factors by him. He did not actually see the heredity particles, but the particles did explain the pattern of inheritance shown by genetic traits. He maintained that the factors were present in all the cells, somatic as well as germinal cells, and the gametes carried the factors to the next generation. So, gametes form the physical basis or the carriers of heredity. Now, the second point is the chemical basis. So, chemical basis, they say that the heredity is controlled by genes. Now, we say that the heredity is controlled by genes that are definite segments of chromosomes. You know, we have studied genes and chromosomes in class 11th. So, now studying the chemical basis, we can say that these genes and chromosomes, they are particulate bodies. So, the characters travel from the parent to the offspring in the form of genes situated in the chromosomes present in the nuclei of the gametes. The genes are composed of DNA and proteins. The DNA part functions as the genetic material and forms the chemical basis of heredity. Many genes code for proteins, and in this way, a gene manifests itself as some trait of the organism. Genes replicate, and one set of such genes passes into each new cell. So, this is how the offsprings, they inherit the genetic traits from their parents because they are present in each and every cell. Have you understood? So, now we will come down to Mendelism. Now, about Mendel's life first. A little bit about his life, who has contributed so much to genetics. You know, Gregor Johann Mendel, his full name was also known as father of genetics. You know, Gregor Johann Mendel, he was an Austrian born in a peasant family of Moravia from 1822 to 1884. He received school education mainly by his own effort because he belonged to a very poor peasant family, and because of that he had to leave his education also. In between, he became a monk, a religious person in 1843. This slide shows his birthplace as well as the dates of his lifespan, his birth from 1822 to up to his whole life, that is up to his end, that is in 1884. And in 1843 he became a monk in a church. So he was here only, that later on then in 1847 he was made the head, that is the abbot of Augustinian monastery of Saint Thomas at Brunn, Austria, which is now known as Brno in Czechoslovakia. That is the Czech Republic. From here he went in 1851 to the University of Vienna, where he studied natural history and mathematics for 2 years. Do you know that he was the first person to involve mathematics in biology? That is how he made his calculations in biology. So in Vienna, he became interested in the process of hybridization. His choice of subjects and his aptitude for hybridization was very significant in his later work when he worked on the pea plant. Mendel returned to his monastery in 1853. Here he also worked as a teacher of physics and natural history in school. So from 1856 to 1865, Mendel conducted breeding experiments on garden peas in the garden of his monastery. He was not the first person to experiment in the field of inheritance, but he was luckily the first to formulate clear-cut principles or laws of heredity. He published his findings in 1866 in the annual proceedings of Natural History Society of Brunn. His laws form the basis of the science of genetics even till date, and he is always remembered as the father of genetics. But it was very unfortunate that the great significance of Mendel's discovery was not appreciated by contemporary biologists. Why? Because his work was ahead of his time. He published it in a journal that had limited circulation. He was himself not sure of his findings, as he failed to get similar results on hawkweed. He lacked aggressiveness in his personality. His mathematical approach in working out biological problems was strange for the scientists of that time. Mind of the biologist was preoccupied with the Darwin's theory of evolution, and in between all this, Mendel died in 1884 of a kidney disorder without knowing that he had laid the foundation of genetics. So then what happened? Then after that, here you can see Mendel also in the picture. So I hope about Mendel everything is clear to you. You must be wondering why we are talking about all this, but students, this is very important before we come to the actual Mendel's laws. So I told you that Mendel died without knowing that he had laid the foundation of genetics. His work was rediscovered. What happened? In 1900, his results were rediscovered through hybridization experiments conducted independently by three biologists, namely Hagodi Veris of Netherlands, Karl Collins of Germany, and Erich von Schoenmark of Austria. These scientists deserve all the credit for naming them after Mendel and for bringing them to the notice of the biologists, because Mendel's work got unnoticed first, right? Then Mendel's work was republished by De Vries in Flora in 1901. A few years later, W. Bateson and others confirmed Mendel's work and found that the same laws applied to animals also. Which laws? The laws which he proposed in pea plant. Okay. So now we come down to the pea plant which was taken up by Mendel. Mendel took pea plant. He was very lucky enough to take it as his experimental plant, because the plant he chose had direct characters like dominant, recessiveness. He was not he did not cover the characters which he took, did not have any complications in them. So here on your screens are displayed these characters which were taken up by Mendel. These are the 7 pairs of the contrasting characters in pea plant which was studied by Mendel. We will talk about them now. So as far as the seed is concerned, the form of the seed, round or wrinkled, then the color, this cotyledons whether yellow or green, then the flower color whether white or violet. As far as the pod was concerned, that is full pod or the constricted pod, yellow pod or the green pod. And from the stem he took the axial pods, that is the flowering, either the pods were formed axially or they were formed in terminal position, whether the plant stem was long or it was short, that is the tall plant or a dwarf plant. So we'll take these now. What were the advantages of selecting this plant? He worked on pea plant which is known as Pisum sativum. The name of the pea plant is Pisum sativum. So the basis for selecting this plant were because of some of its special advantages, which were like, firstly, the garden peas were found to differ in certain definite and easily detectable traits. That is, some of the plants they were tall and climbing, whereas others were short and bushy. Some had colored flowers, violet color, some white flowers, some had round seeds, some had wrinkled seeds. So Mendel noted 7 pairs of such contrasting characters, which I have just mentioned and are displayed on your screens. The traits which always appear in 2 opposing conditions, that is one dominant and the other recessive. That is what we call as a contrasting trait. A dominant means, do you know the meaning of the word dominant? Which will show its own presence, and even if their other one is present, that is the recessive trait is present. In the presence of the recessive trait also it shows its own presence, whereas the recessive trait is the one which only expresses itself in the absence of the dominant. Geneticists use the term character for a heritable feature such as flower color that varies among the individuals. Each variant for a character such as purple or white color of flowers is known as a trait. So these are some of the terminologies which I am telling you. The trait of each kind of pea plant were preserved in generation after generation because the plant had bisexual flowers and normally resorted to self-pollination. This is very important. The pea plant has bisexual flowers and has self-pollination naturally. Complete enclosure of the reproductive parts by petals ensures self-pollination. A plant bearing bisexual self-pollinated flowers is more both male and the female parent to its own seeds. Right? What is a self-pollinating flower? This you know, that a plant bearing bisexual, that is both the male and the female parts, and they pollinate to make its own seeds. So over generations, when this process is carried over, it leads to genetic uniformity up to a large extent. Then I'll show you one more diagram, that is how to make hybridization or cross pollination in pea plant. Here you see the pea plant, and then you see that with scissors the process of emasculation being done, that is, they are being removed. I will focus it over here. Here you see the plant with all its parts, the petals, the carpel, you see the stigma, the anther, the stamen, male and the female parts. Now if we want to make a hybrid, that is cross pollinate the plant, then before the maturity of the plant, these anthers, they are removed, right? Here you see the male and the female parent. So from the female parent, the anthers, they are removed before maturity so that they should not pollinate its own flower, and artificially from another flower which is taken as the male parent, with the help of a brush, the pollination is done artificially, right? So artificial cross pollination here. So artificial cross pollination, that is hybridization, it is achieved by removing the stamens. The process is known as emasculation before the pollen grains are matured, right, from the female flower, and then they are dusted with the desired pollen of the male plant, which we select as the male parent. So here you can see all these steps in the diagram. Then the pea plant has one more advantage, that is, being annual, it had a short life cycle, so that the results can be seen within a year. Pea plant produced many seeds in one generation. This helps in drawing correct conclusions because we need mass numbers in order to reach the exact answer or the correct conclusion. Then pea plant can be raised, maintained and handled very easily. So Mendel worked on only pure characters or the true breeding traits, which means that a plant on self-pollination produces offsprings with the same trait. Like for example, a plant with 4 purple flowers is true breeding if its seeds raised by self-pollination, they all produce purple flowers and no other color. So this is achieved by self-pollination. Self-pollination leads to the purity of the traits. Pea plants having each of them 7 characters he selected were very easily available. So these were his own ideas or in his own observations which made him select Pisum sativum as his experimental plant. Now the Mendel's experimental technique. We talked about the material, that is the pea plant. Now what was his technique? We can divide his technique in 3 points which we are going to take up. That is selection of pure parent plants, hybridization of pure plants and self-pollination in hybrids. We will discuss these one by one. So first is the selection of pure parent plants. Mendel first confirmed that the trait of 7 characters noted by him were true breeding. That is, they were passed on from parents to offspring. For this, he raised seeds by self-pollination for pea plants having different traits and sowed them in his garden. The plants that grew from these seeds showed that each trait of 7 characters were handed down by the parents to the offsprings, that is, they were pure. Seeds from tall plants produced all tall plants, and yellow seeds produced plants with yellow seeds, and similarly same with all other characters which he took into consideration. So Mendel called each of these as a pure trait. So a pure trait is that trait which is passed on to 100% of the offsprings from the parent as such. Next is the hybridization of pure plants. Now crossing or mating of these two varieties of plants is known as hybridization. That means when we take pure breeding plants, so the pure breeding plants which were used by Mendel in his experiment, these hybridization experiments, were like tall and dwarf. He took pure tall and pure dwarf. That is for the size of the plant. Flowers of one plant, say tall plant, they were emasculated, and pollens from flowers of another plant, say dwarf, were dusted over the stigma of the former. That is the tall plant. So the flowers to be pollinated by hand were kept enclosed in paper bags from the very beginning to avoid entry of unwanted pollen. That is, we have to be sure about what we are doing, has to be 100% foolproof. So the seeds formed by crossing the plants were collected and plants raised from them the next year. That means seeds were formed this year, seeds were kept, then they were sowed the next year. So whatever we are talking about the Mendel's laws, and it's not in a minute but we just talk about them and the whole process is complete. It takes time. That is, firstly you have to be sure that the plants which you are taking, they are pure parents, they are true or pure to the character which you are going to study. Next, when you perform the hybridization experiment, then it should be properly carried out, and the seeds will be formed at the end of the season, and those seeds will be kept and sowed in the next season, right? So these seeds, they will be sown the next year, and they will produce hybrid plants that constitutes the first generation. So the first generation of Mendel's crossed was named as the first filial generation, indicated by the symbol which we use even today, that is F1, right? So this is indicated by the symbol F1 by Batson and Saunders in 1902. Filial means the offspring. So first filial generation or the first offsprings. Then comes the self-pollination in the hybrids. This will form the next generation, that is the F2 or the next filial generation. So the hybrid plants of F1 generation, they will form seeds, right? They will be subjected to self-pollination. We will not perform the hybridization experiment here again. They will be allowed to self-pollinate, which a pea plant does naturally. But to be extra sure, we can enclose the flowers in paper bags so that no other foreign pollen comes near it. So the seeds produced, they are collected and then sown again the next year. They produce the second generation, which is known as second filial generation. This is indicated by the symbol F2. Self-pollination in F2 gives rise to third generation, right? Where we will refer to it as F3, and we can continue with the generations so on. But today we will stop here, and there are certain terms in this genetics and the Mendelian genetics which you should understand before we proceed further with the actual experiments and their results. So students, I have mentioned a few of them here, and I have mentioned the site also from where you can visualize more terms. And if any case of any difficulty, please contact me and I'll get it clarified. Thank you. Hope you understood it well. So till tomorrow, stay safe, stay healthy.